Array or DNA chip is a special array of oligonucleotide probes closely arranged on a small solid support surface. The oligonucleotide probes represent the nucleotide sequence in known gene are synthesized on the solid surface in such a way that both the position and the nucleotide sequence of each probe is microarrays can be used to identify organisms and the specific gene by hybridization of DNA from the organism to the oligonucleotide probes in the microarray. There are several ways to make microarray. The one described here involves methodology adapted from computer chip technology. Piece of glass about 1.5 by 1.5 centimeter is covered with a mask containing holes corresponding to the site of attachment of the desired nucleotides. The first nucleotide to be attached is added and made to bind by a light activation reaction. The mask is then replaced by another mask which exposes sites for addition of the next nucleotide. This process is repeated until all of the oligonucleotide probes, each with known sequence and position, is synthesized. Although only 5 nucleotides are known in the nucleotide of this animation, they are usually 15 to 25 nucleotides in length in the microarray. The DNA from the organism being studied is isolated, fragmented, and tagged with a fluorescent dye. The DNA fragments are then incubated with the chip. DNA from the organisms that is complementary to the DNA in the microarray bind together and the unbound DNA is washed away. The surface of the microarray is then scanned with a laser beam and the data obtained is used to produce a visible image. Color intensity indicates the extent of hybridization to the different probes. Over 200,000 probes can be constructed on the single microarray. With this technology, and desired sequence of nucleotide can be constructed to form a probe. In the presence of gene for antibiotic resistance, another trait can easily be determined, and gene expression can be monitored by hybridizing complementary DNA obtained from a single